Dear viewers, today let us discuss about one more very interesting topic from the chapter Sexual Reproduction in Flowering Plants, that is pollen-pistil interaction. Before moving on to the topic, let me remind you what we have discussed in the previous classes. So we have discussed about what exactly do we mean by pollination and what are the different types of pollinating agents or the pollinators. Let me say, for instance, we have discussed about the pollination by wind, that is anemophily, by water hydrophily, and by animals, different ways we have studied that pollinating agents, that is we refer it to as zoophily. So here, when we come to the topic pollen-pistil interaction, let me tell you, my dear friends, that during the process of pollination, many pollen grains from different flowers of different plants may come and fall onto the stigma of a particular plant's flower. So it does not guarantee us the right type of pollen grain will deposit every time onto the receptive stigma of a particular flower. Many different pollen grains may fall, so definitely it will happen. So there comes two important aspects. The flower either it has to accept or it has to reject. So we use two different terms here to talk about the pollen-pistil interaction. First we talk about the compatibility. Secondly, we talk about incompatibility. Compatibility refers to something which is easy or capable of interaction. Easy to interact or capable of interaction is what we term it as compatible. Incompatible, which is not easy to interact or which is not capable of interaction, is incompatible. So here, if the pollen of a particular species which belongs to the flowers, that particular flower where it to its stigma it is falling, then it will accept. So then we can say it as compatible. If the pollen grain is from some other flower which does not belong to that particular species, definitely it will not accept. So then we can say that as what? Incompatible. So many important physical factors as well as the chemical factors define pollen-pistil interaction, my dear friends. Many botanists especially have worked on chemical components of flower to understand how compatibility and incompatibility is defined. So let me tell you, for instance, say for example, we have with respect to the pollen grains, for example, let us consider, so exine has sporopollenin. Then let us say for example we have germ pore, so which consists of allergens. Then it has certain enzymes and also to go with certain proteins and lipids. So this is one important aspect. Next, let us say for example we have intine. So intine consists of both the carbohydrates, as we all know that it is generally made up of pectin and cellulose, so obvious that it will have carbohydrates. Together with that it also has proteins. And other than the intine, let us also consider the stigma. Stigma consists of carbohydrates, proteins as well as lipids. So all these chemical factors which are present here with respect to the different parts of the pollen grain and the flower will decide the compatibility or the incompatibility, whether the pollen grain when it falls onto the stigma will accept or reject. That can be decided. So this is the botanists, very recently they have in fact discovered this chemical interactive nature to decide pollen-pistil interaction. My dear friends, further let me tell you, we have, for example, if it is compatible, it will accept. So the pollen grain after falling onto the stigma will start to germinate. So when it germinates, it will start to form the pollen tube. So it will start to form the pollen tube. This pollen tube has to pass through the style, isn't it? We know that. So when it has to pass through, it needs passage for its movement. So there certain important enzymes we have for example pectinases and hydrolytic enzymes which are released by the pollen tube, so that there is a very clear and easy passage for the pollen tube to move across inside the style. Remember this point. So furthermore, my dear friends, let me tell you that the pollen tube, okay, after it is compatible, it will secrete the pectinases and hydrolytic enzymes. It will rupture the style and it will move. Okay, after its movement, the pollen tube has to enter the embryo sac for the further fertilization process to occur. For that to happen, the pollen tube can enter the embryo sac in three different manners. Firstly, one we have is porogamy. So the pollen tube can enter directly through the micropyle. So this is we call porogamy. Okay, example we can bring in here is, for example, we have lily. So next we have chalazogamy. So here the pollen tube entry is through chalaza. This is the chalaza. So I have drawn here all anatropous ovules are inverted ovules. So this is the chalaza, will become the basal part of this anatropous ovule here. So from this also the pollen tube can enter, or this is the micropyle. Through micropyle it can enter. So as I said, through micropyle the entry of pollen tube is referred to as porogamy. Through chalaza if it is entering, then we refer that to as chalazogamy. As in the case of casuarina we can observe. Then we also have mesogamy. So we have the integuments here, that is the walls of the ovule. Through this it can enter and get inside the embryo sac and further undergo the process of fertilization, as we can observe with respect to cascata. So these are the three different ways through which the pollen tube can enter. And let me tell you, my dear friends, other than this, now finally it is also, we already know that the pollen grains also in fact shed at either two cell stage, that is the vegetative cell and the generative cell stage, or it can shed at the three cell stage also, where in fact generative cell will divide to form two male gametes, so it becomes three cell stage. So anyway, so these aspects pertaining to pollen-pistil interaction we need to remember. And finally, if we need to frame the definition for pollen-pistil interaction, we can say as such, for example, let us consider this as the stigma, style, ovary. Okay, thalamus, fine. The coming and falling of the pollen grain onto the stigma, and then formation of the pollen tube. Okay, so then the pollen tube's entry into the ovule. Okay, until the fertilization, all the changes, which occurs are referred to as pollen-pistil interaction. So these are few important aspects today we have discussed. So I request everyone to understand here the compatibility, that is how the pollen can interact with the stigma, or whether it is generally in case of compatibility it is accepted, whereas with respect to incompatibility it is rejected. So these interactions are very much necessary, my dear friends. To go with that, please also remember the chemical components which are necessary for deciding this compatibility and incompatibility, and how the pollen tube enters through the style. So remember pectinases and hydrolytic enzymes which are necessary to tear the style, tissues of the style. So further also remember how the pollen tube can enter through the ovule into the embryo sac. Either it can be porogamy, chalazogamy, or mesogamy. So remember few examples for the same. So this all in fact completes the topic pollen-pistil interaction. Keep updated for more videos. Definitely you will be benefiting with the other coming topics. So I request everyone to like, subscribe and keep supporting. So thank you very much.